अस्सलाम वालेकुम स्टूडेंट्स। होपफुली यू आर फाइन। कल हम लोगों ने होम्योस्टेसिस डिस्कस किया था। टुडे वी आर गोइंग टू डिस्कस योगिया साइकिल। व्हाट्स योगिया साइकिल? In eukaryotic organisms, the ammonia deposited in the mitochondria of the hepatocytes that is converted to urea in the urea cycle. Urea cycle was discovered by Krebs Hanslet in 1932. Therefore, urea cycle is also called Krebs Hanslet cycle, and it's also called ornithine cycle because the first member of this reaction is called ornithine. Therefore, this is called ornithine cycle. Urea is synthesized in the liver, and from the liver it is transported to the kidney. When we discuss the urea cycle, that is begin inside the liver mitochondrion or its mitochondria, and in short form the mitochondria is also called mitose. But there are some steps, like three steps, that is taken place in the cytosol of cells. Therefore, ammonia, two molecule of the ammonia combines with one molecule of the carbon dioxide to produced by mitochondrial respiration to form carbonyl phosphate in the matrix. The urea cycle, this is a reaction which is dependent ATP, or we can call it as ATP dependent reaction, where some enzymes like carbonyl phosphate synthetase are used. Carbonyl phosphate synthetase 2 is also used for this cytosolic form separate function in the pyrimidine biosynthesis. Carbonyl phosphate, it is an activated carbonyl group donor which enter into the urea cycle. When we discuss the steps of the urea cycle, the first step of the urea cycle is the carbonyl phosphate donates its carbonyl group to ornithine to form citrulline. With the release of phosphate group, one phosphate group is released during this reaction. This reaction is catalyzed by enzyme which is known as ornithine transcarbamylase. Citrulline passes from mitochondrion to the cytosol of the liver cells. The second phase, the second step of the mitochondrion, the second step of the urea cycle is when the amino group enter from aspartate, which is generated in the mitochondrion by transamination and transported into the cytosol by a condensation reaction between the amino group of aspartate and carbonyl group of citrulline, forming arginosuccinate. It is a cytosolic reaction which is catalyzed by enzyme known as arginosuccinate synthetase. Dear students, one thing should be confirmed, that all the enzymes, they are according to their substrate, like arginosuccinate, yes, and arginase, urease, and there are many other enzymes. They are mostly enzymes, they are according to their substrate. We classify these enzymes, or we give a name to these enzymes according to their substrate. Come to the topic. The third phase, that is the arginosuccinate, is then cleaved by arginosuccinase to form arginine and fumarate. Two compounds or two molecules are come, that is arginine and fumarate. Now, the fumarate can enter to the mitochondria and join into the citric acid cycle. It is only reversible step in urea cycle. Come to the fourth, the cytosolic enzyme, that is arginase. This arginase divide the arginine to urea and ornithine. Ornithine come to the first and run the cycle again. Therefore, I told you people that it's also called ornithine cycle. This is the diagram. I'm really sorry that after deamination of the amino acid, what is deamination? When the amino acid divide, or we can call the amino group is separated from the amino acid, it's called deamination. Here you people आपको मैंने बताया था, I told you earlier that two molecule of the amino group, while in this pic you people see that only N is clear, but H3 it's not clear. I'm really sorry, due to some problem I can not take a clear pic. Therefore, you people can see this pic in the second year biology on page number 16, where you people can clearly see this pic. Now, the all this pic I explain in the previous slides. Therefore, you people can see in detail. Thank you. Allah Hafiz. We will discuss the kidney in next slides. Allah Hafiz.